So it's my great pleasure to introduce Jaime Louro today. So Jaime started studying pharmaceutical sciences, and then he briefly worked as a pharmacist before going back to the lab as a trainee at the Institute for Molecular and Cell Biology in Oporto. He then moved to the Universidad Autónoma de Madrid for a PhD in molecular biosciences. That led to two successful postdocs in structural biology, one at the IGBMC in Strasbourg and one at the University of California with Professor Andres Lechner, where he worked on the project that he will be presenting today, which is entitled Structural Basis of Nucleosome Recognition by Conserved, Dsup, and HMGN Nucleosome-Binding Motifs. Since the beginning of the year, he's back in Europe on Spanish National Cancer Research in Madrid. So welcome back to Europe, Jim. We're looking forward to your talk, and the floor is yours.
Yeah, I want to thank the organizers. It's a great opportunity to be here to disseminate my work. So today you will hear about how two different proteins, Dsup and HMGN2, which have a conserved nucleosome binding motif, recognize the nucleosome. So first I need to introduce tardigrades, also known as water bears or moss piglets. They are, to be active, they just need a thin film of water. And then in adverse conditions, they curl up and they enter this low metabolic state, cryptobiosis. So scientists are still trying to understand what makes tardigrades so resistant. And today I'll talk about a protein that has something to do with it. So that protein is Dsup, damage suppressor, and protects DNA from damage, hydroxyl radicals, irradiation, double-strand breaks. And our collaborators found that this protein, so it's a invertebrate animal, right? So this protein has a nucleosome binding motif. If we take a look at the amino acid composition, it suggests intrinsic disorder. It makes sense, right? If you need to resist very extreme conditions, the proteins are already unfolded. Let's put it like this. Dsup, if it is transfected into several eukaryotic cells, it protects those cells from damage. And because of this, it might have a potential as a model organism for radio tolerance studies or even biomedical applications, like, for instance, in cancer patients undergoing radiotherapy. So we asked ourselves, how does Dsup specifically recognize nucleosomes? And for that, we used cryo-electron microscopy. So our collaborators assembled 601 nucleosomes, and they incubated them with a molar excess of Dsup. We had to chemically fix the sample with glutaraldehyde, so lysine-lysine cross-linking. And only after that treatment, we could see Dsup bound to the nucleosome. So I won't get into details. We had very good resolution in our structures, and we saw two molecules of Dsup bound to opposite faces of the nucleosome. Now I'll zoom in one molecule so we can take a look at the interface between Dsup and the nucleosome. So again, there are two main amino acids in Dsup responsible for the interaction. So one arginine 367 and arginine 363, inserting into two cavities of the H2A H2B dimer. If you see, all residues in H2A H2B dimer are acidic, except for this glutamine 47. The take-home message here is that two arginines anchor into the acidic patch. So we had high resolution for the part contacting the acidic patch, but since Dsup is 43 kilodalton, is relatively a relatively large protein, we were looking for other densities. And indeed, we obtained low resolution densities near H3 alpha N and near in the vicinity of alpha 1 of H3, the other copy of H3, as well as the loop 1. These are also hotspots for nucleosome binding by other proteins. We obtained other classes. So it was cool, to my knowledge, for the first time to see Dsup. But since the classes are low resolution, we just know what was already known, right? It's very flexible with multiple conformations around the nucleosome. We believe that this is related to the DNA DNA damage protection role because these fuzzy interactions shield the DNA. Then we set out to study HMGN2. I'll introduce this protein later, but now I'll jump into the structure. So we used 5S ribosomal DNA nucleosomes, not the synthetic 601. And again, we had to cross-link the sample. HMGN2 resembles this up, right? Two molecules bound to opposite faces of the nucleosome. Now let me zoom in. It will become clear in a moment why this time I'm zooming in on two molecules and not just one. But first, what's common to this up? Again, two arginines, as you can see here. Oh, sorry. Two arginines inserting into the acidic patch into two cavities. But I must explain what's the link between these two proteins. So we have damage suppressor from an invertebrate animal, and we have a high mobility group nucleosome binding to protein from vertebrates. What's the link? The link comes from our collaborators. They noticed that there is a nucleosome binding motif that if we align, I hope you can appreciate how similar they are. So in primates, actually, there are 5 members of this family, and they all have the same nucleosome binding motif. So I think the conclusions we draw for HMGN2 most likely can be expanded to the other members of the family. So HMGN2 in humans is not giving us superpowers to resist radiation. No, it's not that. It's involved in transcription regulation, DNA damage repair, and also cell differentiation. And it also has, at the level of amino acid sequence, it has a tendency to be intrinsically disordered. So are there differences in how HMGN2 and Dsup recognize our nucleosomes? Yes, we saw differences. So here I'm overlaying the two H2A, H2B dimers from opposite faces of the nucleosome. I hope this makes sense. And you can appreciate that the way the two molecules bind the acidic patch is not exactly the same. There are some asymmetries. So here in black, you have the interactions that are common to the two molecules, and in magenta, you have the interactions that only exist in one of the molecules. We also saw asymmetries in the side chains of the acidic patch and of this glutamine. For instance, here is pointing in and pointing out, and here the same. So yeah, don't ask me exactly what this means, but it's one observation, and we wanted to report it at the molecular level. The second difference, and now I'm using a scheme here, I'll try to explain. So this arginine in both HMGN2 and Dsup, and I'm talking about this arginine here. So it's the same in the sense that it's the same register, but the arginine that inserts below, let me go back, the arginine that inserts into this cavity here, so it has a mismatched register. So the binding is not exactly the same. And here, this nomenclature for arginine anchor and type 1 arginine comes from McGinty and Tan review on nucleosomes. Yeah, I forgot to mention before that this binding mode of proteins with arginines anchoring to acidic patch has been seen already many times before. The first time was in a work by Caroline Lager with a LANA peptide from a virus. So then we set out to analyze nucleosomes containing the 5S ribosomal DNA sequence. So let me introduce this gene. It is transcribed by RNA polymerase 3. Of course, it needs transcription factors. I won't get into that. What's special about this gene is that it contains an internal promoter. It's right in the middle of the gene. So our nucleosome has the first element of the internal promoter, has a transcription start site, and also all the upstream region. Our position 0, the diad of the nucleosome, would be roughly here. So, and why is it important to solve a structure of a nucleosome alone? So, because it is a natural DNA and not a synthetic one like the 601, because already in 1985 it was discovered that this DNA could position nucleosomes. 2 years later, the box A of the promoter was discovered. And then in 1988, nucleosomes could be assembled. But almost 40 years passed and no structure yet. So yeah, it's important. So we determined 3 structures from the same sample, 3 structures. Let me explain better. So the first one is the canonical nucleosome because the 2 DNA ends are wrapped around the histone octamer. Don't get confused. In red, I represent absence of signal, absence of electron microscopy density, meaning if we don't see density, meaning that there is flexibility. So in other words, our DNA was 167 base pairs. It has 10 base pair linkers from each side that we don't see because of the flexibility. We obtained 2 more structures in which the flexibility of the DNA ends was increased. And in this structure in particular, from both sides. So again, I won't get into details, but because our maps were good with enough detail, we could model the DNA in the correct orientation. I hope this makes sense. The nucleosomes are pseudo-symmetric, so you can model the DNA in one orientation or 180 degrees rotated. And we managed for each structure to find the orientation, and because of that, I know where the upstream DNA would be and the downstream would be. They are flexible, but I think you understand what I mean. And also because of that, I know where the transcription start site is, and I know where box A would be. I mean, it's moving, but it would be on this side. Other important piece of information is the percentage of particles present in our structures coming from the same sample. So most of the particles are unwrapped. So altogether, I think we can state that the 5S nucleosome preferentially unwraps through the downstream side, exposing the box A, the internal promoter, in vitro at the mononucleosome level, of course. Finally, we asked if cross-linking affected our 5S ribosomal DNA nucleosomes. So our collaborators assembled the nucleosomes and they split it in 2 pools. One pool was not cross-linked, and the other pool was cross-linked with 2 types of cross-linkers, either glutaraldehyde or formaldehyde. And to our surprise, we obtained a single structure. It's not that the nucleosome was larger. No, we obtained a single structure. And also, very importantly, this structure does not match any of the non-cross-linked structures. So the difference is the flexibility in the DNA ends is much higher. I hope you can appreciate it. Here indicated by the red surface. So in other words, cross-linking the nucleosomes induce an artifactual state with increasing breathing of our 5S nucleosomes. So today I showed you that our 5S nucleosomes preferentially unwrap through the downstream side and cross-linking with 2 types of cross-linkers induces DNA end flexibility. And I showed you that 2 molecules of tardigrade Dsup and vertebrate HMGN2 using mainly 2 arginines can occupy both nucleosomal acidic patches. So this could shed light into the function of these proteins The binding of the nucleosome to the nucleosome data from other labs there is cooperativity. So it's possible that these nucleosomes have these proteins have an important role in gene regulation because they occupy both acidic paths, impeding, for instance, chromatin remodelers of binding to the nucleosome. This is just speculation. So I want to thank my mentor, Andres Leshner, also our collaborators in Kadonaga Lab. Grisel prepared all nucleosomes that you saw today. I did all the cryo-EM. We want to thank also NIH for the funding, George Casavetes, the physics, computing, and cryo-EM facility at UCSD, and I'm personally grateful to the J1 visa program. My name is Jaime. I'll take your questions. Thank you.
Thank you, Jim. Very, very nice linear talk. And of course, love the tardigrades every time I see them. While we wait for question coming in, I think Ben had one.
Yes, great talk. So with the second part, I was struck by how much the 5S nucleosome sequence increased the flexibility of the DNA versus the 601. And my understanding, the 5S nucleosome sequence is still a fairly stable, like nucleosome binding sequence. So I'm wondering how much more flexibility or ranges of flexibility would you expect to see if you used additional endogenous sequences?
It's hard to answer. I can only speak for cryo-EM. Even between labs, there is not reproducibility with the 601 sequence. Yeah, so I think there are several pieces of evidence like molecular dynamics, even SACs and other FRET and other techniques that say that the native sequences are much more flexible, but I can't quantify. I don't know. I can only say more flexible. Yeah.
Yeah, it's exciting to see though, like more like I think it's an interesting area to think about in terms of how the nucleosome structure changes with the sequence. So I was quite excited to see your results on that.
Yeah, thank you.
I have a couple of curiosity that are a bit more related to the in vivo situation. So is Dsup expressed also in the proliferative state, or is it specific for the cryptobiosis? And so do actually Dsup and HMGN2 coexist on the genome, or is one and then the other?
So HMGN2 is vertebrate specific.
Okay.
Yeah. The first question, if Dsup is specifically expressed in the cryptobiosis stage, to be honest, I don't know if that is known. Yeah.
Okay.
If it is known, I don't have the answer. Yeah.
And do you know whether that is like carpeting the genome, or there are hotspots of binding for the protection?
Yeah. So I think this the specificity is not given by DNA sequence, but exactly how Dsup knows where to go. Yeah, I don't know.
So it could be possible that it's just covering the whole of the genome to protect the DNA everywhere.
It could be. It could
Okay.
be. Yeah.
Maybe just last question on related to a bit more the gene expression kind of things. Do you know from the structure whether you would have a similar binding with H2A variants like H2AV or similar?
Since the acidic patch is conserved, I don't know about all variants, but since usually they are shorter in the terminals, right? So I expect the same behavior in terms of what we see in our structures, the interface with the nucleosome. Of course, there are many parts of our proteins, HMGN2 and Dsup, that we don't see, and the variants could influence DNA flexibility, and that in turn influence somehow the binding. That's another story. But what we see in our structure should be the same. Yeah.
Okay, fantastic. Thank you very much. So I think we're about time, so I'll pass it to Ben for the next talk. Thanks again, Jamie.